This method can be called ELISA at tissue level to distance itself from the traditional relative semi-quantitative quantification of protein content at cellular and tissue levels, which is the norm in the research lab and in the clinical diagnostic field currently. The main advantages of this technique can be described in 3 words: high throughput, quantitative, and absolute. If we were to add another word, it would be simple.
The implications of this technique extend into the proteomics field, whereas the verification of the biomarkers can be achieved in a high throughput format. This method has broad applications throughout research and clinical field. The quantification of the protein amount at the cellular and tissue levels are still primitive at present.
Generally speaking, individuals new to this method will face no problem learning this technique, and it is quite basic and require minimum lab skills to perform.
We are excited to demonstrate its reliability, simplicity, and accuracy of the technique using the animal model.
The samples can be prepared with any traditional method of sample preparation using any lysis buffers commonly used in the research lab, with or without detergent, like MP40 or Triton X. In our case, we use Triton X lysis buffer. Place a slice of tissue of 5 to 100 milligrams in a 1.5 milliliter tube. Next, add 200 microliters of lysis buffer supplemented with protease and phosphatase inhibitors. Next, homogenize the tissue with a homogenizer for 1 minute on ice. Then, centrifuge the sample for 10 minutes at 8,000 times G at 4 degrees.